In this video, we are going to learn about integrins, interaction of cells with extracellular materials. It was noted in the previous discussion that components of the ECM such as fibronectin, laminin, proteoglycans, and collagen are capable of binding to receptors situated on the cell surface. The most important family of receptors that attach cells to their extracellular microenvironment is the integrins. What is so peculiar about integrins? They attach cells to the extracellular microenvironment. But earlier we have learned about components of ECM, extracellular matrix, such as fibronectin, laminin, proteoglycans, and collagen, which are capable of binding to receptors situated on the cell surface. Okay, so integrins are a family of membrane proteins found only in animals that play a key role in integrating the extracellular and intracellular environment. On the outer side of the plasma membrane, integrins bind to a remarkably diverse array of molecules or ligands that are present in the extracellular environment. On the intracellular side of the membrane, integrins interact either directly or indirectly. On the intracellular side of the membrane, integrins interact either directly or indirectly with dozens of different proteins to influence the course of events within the cell. Integrins are composed of two membrane-spinning polypeptide chains, an alpha chain and a beta chain, that are non-covalently linked. 18 different alpha subunits and 8 different beta subunits have been identified. Although more than 100 possible pairing of alpha and beta subunits could theoretically occur, only about 2 dozen different integrins have been identified on the surface of cells with which a specific distribution within the body. Whereas many subunits occur in only a single integrin heterodimer, the beta 1 subunit is found in 12 different integrins and the alpha V subunit in 5 of them. Most cells possess a variety of different integrins, and conversely, most integrins are present on a variety of different cell types. Electron micrographic observations of integrin molecules beginning at the late 1980s suggested that the two subunits are oriented so as to form a globular extracellular head connected to the membrane by a pair of elongated legs. Two subunits are oriented so as to form a globular extracellular head. Extracellular head will be there, which is connected to a membrane by a pair of elongated legs. The legs of each subunit extend through the lipid bilayer as a single transmembrane helix and end in a small cytoplasmic domain of about 20 to 70 amino acids. One exception to this molecular architecture is the beta 4 chain, which has an extra 1000 or so amino acids as part of its cytoplasmic domain. This huge addition makes beta 4 integrins capable of extending much more deeply into the cytoplasm. The first X-ray crystallographic structure of the extracellular portion of an integrin was published in 2001 and displayed a highly unexpected feature. Rather than standing upright as predicted, the integrin alpha V beta 3 was bent dramatically at the knees so as the head faces the outer surface of the plasma membrane. To understand the significance of this bent structure, we need to explore the properties of integrins. Many integrins, including the ones can exist on the surface of cell in an inactive conformation. These integrins can be activated. These integrins can be activated rapidly by events within the cell that alter the conformation of the cytoplasmic domains of the integrins subunit. Changes in conformation of the cytoplasmic domains, many integrins, including the ones shown, can exist on the surface of a cell in an inactive conformation. These integrins can be activated rapidly by events within the cell that alter the conformation of the cytoplasmic domains of the integrin subunits. Changes in conformation of the cytoplasmic domains are propagated through the molecule increasing the integrin's affinity for an extracellular ligand. For example, the aggregation of platelets during blood clotting occurs only after the cytoplasmic activation of alpha 2b beta 3 integrins, which increase their affinity for fibrinogen. This type of alteration in integrin affinity triggered by changes occurring inside the cell is referred to as the inside out signaling. In the absence of an inside out signaling, the integrin remains in the inactive state, which protects the body against the formation of an inappropriate blood clot. A type of evidence, a body of evidence A body of evidence strongly suggests that the bent conformation of an integrin corresponds to the inactive state of the protein, one that is incapable of binding a ligand. In fact, when an alpha V beta 3 integrin containing a bound ligand is analyzed, the integrin no longer exhibits the bent structure but is present instead in an upright conformation as illustrated. In fact, when an alpha V beta 3 integrin containing a bound ligand is analyzed, the integrin no longer exhibits the bent structure but is present instead in an upright conformation. The ligand is bound to the head of the integrin in a cleft where the alpha and beta subunits come together. If the bent and upright structure represent the inactive and active states of an integrin, respectively, then it is important to consider what type of stimulus is capable of triggering such a remarkable transformation in protein conformation. In the bent inactive conformation, the transmembrane domains of the two subunits are in close proximity, apparently held together by non-covalent interactions between residues of the two helices. The cytoplasmic domains of integrins bind a wide array of proteins. One of these proteins, called the talin, causes separation of the alpha and beta subunits as shown in the structure of activated integrin and the schematic model in the figure. Mutations in talin that block its interaction with beta integrin subunits also prevent activation of integrins and adhesion to the ECM. Separation of the cytoplasmic tails and transmembrane domains of an integrin that results when talin is bound is thought to send a in change in conformation through the legs of the integrin, causing the molecule to assume an upright position in which the head of the protein is capable of interacting specifically with its appropriate head ligand. An increase in affinity of individual integrins is often followed by clustering of the activated integrins, which greatly enhance the overall strength of the interaction between the cell surface and its extracellular ligands. Integrins have been implicated in two major types of activities: adhesion of cells to their substratum or to other cells, and transmission of signals between the external environment and the cell interior, which are the two main types of activities by integrins: adhesion of cells to their substratum and transmission of signals between the external environment and the cell interior. We have already discussed the transmission of signals in an inside-out direction, which primarily affects the binding properties of the integrins. Signaling can also occur in the opposite direction, a phenomenon known as the outside-in signaling. The binding of the extracellular domain of an integrin to a ligand, such as fibronectin or collagen, can induce a conformational change at the opposite cytoplasmic end of the integrin, especially its beta subunit. Changes at the cytoplasmic end can in turn alter the way the integrin interacts with a host of different cytoplasmic proteins, modifying their activity. For example, outside in signals can induce a conformational change in talin on the inside of the membrane, initiating a cascade of events leading to the polymerization of actin filaments of the cytoskeleton. Similarly, binding of integrins to an extracellular ligand can trigger the activation of cytoplasmic protein kinases such as FAK and SRC. These kinases can then phosphorylate other proteins, initiating a chain reaction. This kinases can then phosphorylate other proteins, initiating a chain reaction. In some cases, the chain reaction leads all the way to the nucleus where a specific group of genes may be activated. Outside-in signals transmitted by integrins and other cell surface molecules can influence many aspects of cell behavior, including differentiation, motility, growth, and even the survival of the cell. The influence of the integrins on cell survival is best illustrated by comparing normal and malignant cells. Most malignant cells are capable of growing while suspended in a liquid culture medium. Normal cells, in contrast, can only grow and divide if they are cultured on a solid substratum. If they are placed on suspension culture, they die. Normal cells are thought to die in suspension culture because their integrins are not able to interact with extracellular substances and as a result are not able to transmit life-saving signals to the interior of the cell. When cells become malignant, their survival no longer depends on integrin binding. The role of integrins in transmitting signals is a very active area of research and will be explored further later. The role of integrins in transmitting signals in a very active area of research. Linkage between integrins and these ligands mediate adhesion between cells and their environment. Because individual cells may express a variety of different integrins on their cell surface, such cells are capable of binding to a variety of different extracellular components. Despite the apparent overlap seen in table, most integrins do appear to have unique functions, as knockout mice that lack different integrin subunits exhibit distinct phenotypes. For example, alpha 8 knockouts show kidney defects, and alpha 4 knockouts exhibit heart defects, and alpha 5 knockouts display vascular defects. Many of the extracellular proteins that bind to integrins do so because they contain the amino acid sequence arginine glycine aspartic acid, or in abbreviated amino acid nomenclature RGD. This tripeptide is present in the cell binding sites of proteoglycans, fibronectin, laminin, and various other extracellular proteins. The cell binding domain of fibronectin, which is extended RGD containing loop, can be seen. The diversity and the discovery of the importance of the RGD sequence has opened the door to new avenue for treatment of medical conditions that involve receptor ligand interactions. When the wall of blood vessel is injured, the damaged region is sealed by a controlled aggregation of blood platelets, which are non-nucleated cell fragments that circulate in the blood. When it occurs at an inappropriate time or place, the aggregation of platelets can form a potentially dangerous blood clot, thrombus, that can block the flow of blood to major organs and is one of the leading causes of heart attack and stroke. The discovery of the importance of RGD sequence has opened the door to new avenues for treatment of medical conditions that involve receptor ligand interactions. When the wall of blood vessel is injured, the damaged region is sealed by the controlled aggregation of blood platelets, which are non-nucleated cell fragments that circulate in the blood. When it occurs in an inappropriate time or place, the aggregation of platelets can form a potentially dangerous blood clot, thrombus, that can block the flow of blood to major organs and is one of the leading causes of heart attack and stroke. The aggregation of platelets required the interaction of the platelet-specific integrin and soluble RGD containing blood proteins such as fibrinogen and von Willebrand factor, which act as linkers that hold the platelets together. Experiments with animals had indicated that RGD containing peptides can inhibit blood clot formation by preventing the platelet integrin from binding to blood proteins. Experiments with animals had indicated that RGD containing peptides can inhibit blood clot formation. This finding led to the design of a new class of antithrombic agents, agrastat and integralil, that resemble the RGD structure but bind selectively to the platelet integrin. Both of these drugs are based on the structure of compounds present in snake venom. A specific antibody, ProRio, directed against the RGD binding site of alpha 2b beta 3 integrins, can also prevent blood clots in certain patients undergoing high-risk vascular surgeries. A different integrin targeting antibody Tisabri is prescribed for treatment of multiple sclerosis. The cytoplasmic domains of integrins contain binding sites for a variety of cytoplasmic proteins, including several that act as adapters that link the integrin to actin filaments of the cytoskeleton. The cytoplasmic domains of integrins contain binding sites for a variety of cytoplasmic proteins, including several that act as adapters that link the integrin to actin filaments of the cytoskeleton. The role of integrins in making the connection between the ECM and the cytoskeleton is best seen in two specialized structures, focal adhesions and hemidesmosomes. We can see a model of integral activation, schematic representation of heterotrimeric integrin molecules in the bent inactive conformation and the upright active conformation, right. The switch in conformation is triggered by the binding of a protein, in this case talin, to the small cytoplasmic domain of the beta subunit. Binding of talin introduces a separation of the two subunits and conversion to the active conformation. Activated integrins typically become clustered as the result of interactions of their cytoplasmic domains with the underlying cytoskeleton as indicated. The domain structure of each subunit, seen in the ribbon drawings, is shown by using globular structure segments. The extracellular ligand, in this case is a collagen fiber, binds to both of the subunits in the head region of the activated integrin dimer. Cytoplasmic tails can be seen, alpha and beta parts. Collagen will be there and talin is attached with it. So alpha 2b beta 3 integrin, blood vessel wall platelet, platelet aggregation, fibrinogen and platelet addition along with RGD peptides. The role of integrins in platelet aggregation. Blood clots form when platelets adhere to one another through fibrinogen bridges that bind to the platelet integrins. The presence of synthetic RGD peptides can inhibit blood clot formation by competing with fibrinogen molecules for the RGD binding sites on plasma alpha B2B beta 3 integrins. Non-peptide RGD analogues and anti-integrin antibodies can act in a similar way to prevent clot formation in high-risk patients.